So I am so excited today. As I mentioned, this has been a fan favorite in the past, and we have the conversation back. It's with Drs. Kim Chapman and Brian Wamoff, and this is a academic medicine researcher and someone in industry, and we're going to have to listen into their conversation about how they've moved forward clinical research in a really fun and interesting way. As all of you know, we do record this, but I also want to be cautious to tell you that we'll send the link out, but it won't be posted to YouTube because we want to be respectful to information that's shared here. So please don't share the link beyond just our group, and then delete the link if you once you've watched it. So we're really excited to have Drs. Chapman and Wamoff with us. And I'm gonna start with Kim Chapman. She's gonna talk a little bit about why she even got involved in this collaboration, just to give us a feel for, as academic medicine researchers, how you could even kind of propose research and think about research from this perspective. If you guys have any questions as we go, feel free to post them as the chat, or you can text me at my cell phone, and I will raise questions as we go. So, Dr. Chapman, thanks so much for joining us. She is literally across a wall from me, so if nothing else, I'll just bang on the wall if something happens. So, Kim, thanks so much for joining us, and it's all yours.
No problem. So, first of all, interrupt us if you have any questions, because although we have, I think, 4 or 5 slides, it's more the story about how I started working with HemoShare and how HemoShare works with me. And part of it is to kind of give everybody a background of what our relationship was like. So let me tell you a little bit about me and my practice and my lab. So I'm a metabolist and a geneticist, and I'm interested in a really rare disease called propionic acidemia. And children with propionic acidemia often die of their disease, and we don't really have very good therapies. And so this is kind of where we started. And a little bit of serendipity is that we met the folks from HemoShare, what is it, Brian, 4 or 5 years ago? Or has it been that long? About 4 or 5 years ago. And so what happened was I had a problem. I had a bunch of patients with a rare disease, and we didn't have a therapy. And HemoShare had this cool technology, and I'll tell you a little bit about this cool technology, although Brian will be able to answer additional questions. And what they could do is they could take liver cells and maintain a liver cell as a liver cell, doing all the right biochemistry, making all the right proteins, so making albumin and cytochrome P450. In addition, maintains all the biochemical pathways. So I have the patients, and HemoShare has a technology. And so we got introduced, and we sat through this meeting, and I kind of talked about what I did, and I suddenly realized that my patients get liver transplants. And they get liver transplants, but their livers get thrown away in the trash for the most part. They're considered medical waste. And all of a sudden, we realized that if we collected those livers, we could suddenly study the disorders and maybe come up with some therapeutics. And so I said we had a biorepository going in our institution, but hadn't thought about collecting explanted organs at that point. But set up and started collecting explanted organs from individuals with rare diseases, and not just my disease, but also some of the other rare diseases that get liver transplant. Now, liver transplant, for the most part, are not cures for these disorders. They just make the disease milder. So what we did is, one of the first patients we collected the liver from, we realized that the liver cells in the HemoShare platform had the normal biochemistry. And as I said, the disorder I'm interested in is propionic acidemia. On the, what would that be, your right side on the slide, propionic acidemia is caused by deficiency of propionyl-CoA carboxylase, or PCC. One of the problems if you have PA is that you block the urea cycle, and so you can have elevated ammonia and decreased urea nitrogen. And in fact, within the HemaSure platform, we were able to demonstrate this, and we published this in 2016. So right now we're looking for a therapy together, and we're looking for therapy together because all of a sudden we have a mechanism to start looking for a therapy. But what we've also discovered is a number of things about the biochemistry for a number of disorders that are treated by liver transplant in my group. So I think this is where we kind of become the discussions, part of the discussion. So the question is, what makes a good partnership between academia and industry? And I know Brian's on there, so chime in whenever you feel necessary. But the first thing I think is equality. Now, equality as an academician with an industry just means that you have to have a back and forth. You have to have a discussion about what each's goals are. Because industry, really, their goal is to produce a product. In academia, your goal is to publish. And so how do you balance those things? I have scientific questions. Their goal is a product. And ultimately, we're going to help each other because that product helps my patients. One of the important things is to understand timeline and to have a big communication about what is the timeline. And timeline is how fast are you going to need information from me, how fast are you going to need products or ideas, or we do genotypes, so genotypes is probably the thing. In addition, what's their timeline for experiments? How fast does things go? We have a publication plan because ultimately, my career is made by publication. They have some other problems or risks, so they can't publish things before they're ready to bring it up to the market, right? So you don't want to expose your company to risk of publishing something too early. Because I think I can safely say, Brian, that HemaSure is not a huge company. And we need to understand each other's goals, and I think this all comes with communication. And that was all of my slides, because I thought at this point we could kind of ask questions and have a discussion. Do you have any thoughts, Brian?
And we're not hearing you, Brian. I'm going to try something.
Me. Okay.
Okay. Now we can hear you. Now talk, Brian. Sorry about that.
Oh, my good. Okay, I had said some things, and I just thought everyone was not interested in what I was saying.
I'm sorry.
So.
Say them again.
I heard nothing, Brian. I heard nothing, Brian. I'm just going, and I'm like, is he still there?
Kim would say something, and then I would say something, and she would just talk right over him. Like, okay, I don't need to speak right now. No, Kim, great job. And, the partnership has been outstanding. And I think one of the things that got it going in the right way from the beginning, and I've been on the other side of these as a former academic at the University of Virginia, where a plan wasn't put in place. And that plan goes all the way back to as data comes out, who owns what, as new molecules are made, for example, in our space, in terms of chemistries, what's the ownership there, and really getting those rules of engagement and those rules that are necessary to move forward in place right from the get-go, or at least when you think there's an opportunity for you to partner with someone in the industry. And Marshall, in particular, as well as the rest of Children's, did a really good job at laying that out. So, in terms of publications, just a few things to highlight. it's not that we don't want to publish. We do publish frequently. We are a small company of 30, a team of 30, but, you know, over the past 10 years, we're now north of 40 publications. So it's not that we don't publish. It's more about when we publish. And I think Kim nailed that on the head. Things need to be timed in a way such that we don't create a competitive advantage against ourselves by putting something out there that someone else could use to either jump by us or use it against us in some other way based on whatever their therapeutic program might be. But there is a lot of value, I will say, in publishing with industry. The standards, and I think I can say this because I've been on both sides of the table now, there is a level of scrutiny that goes into a publication that comes out of industry. And what you'll typically find when you try to publish something with industry is just a lot of it takes time simply because we want to make sure that everything truly is what our academic colleague says it is, all the way down to looking at raw data. And I think for the most part, what you'll see coming out of industry academic publications is the truth. Not that what you see coming out of academic-only publication isn't the truth, but I do feel that, and even as HemoShare publishes with other pharmaceutical companies in different therapeutic areas, there's a level of scrutiny that goes into the data because there's so much at stake when we talk about a potential therapeutic going into the clinic. And any opportunity to negate something in the literature can be devastating for our drug discovery program. So again, it's just another reason why things take time when it comes to publishing.
And there's just so many controls that we run that you would not necessarily run out of your lab. Just so
yeah,
many
no, it's interesting. It's a really good point, Kim. One of the things I've learned, I was in academia from 2005 to 2012 at the University of Virginia in the Department of Medicine, and you do run a lot more controls when you get into industry. And I think it's how you value the data. In academia, we value the data to get it out there, to get conversation going, to advance our research programs, to build your research program. I think it's the same motive in industry. It's just that every piece of data and every claim about something that may be revolutionary or evolutionary in terms of how we impact patients has to almost be bulletproof. But at the end of the day, we fully know that it's science, and it's science done in a lab, and things don't always pan out the same way in other labs. I think we just go to an extra effort to make sure that the opportunity for the data to be repeated somewhere else is there.
Yeah, it's interesting. So when we publish the paper that's actually referred in the slides, it's actually quite fascinating to read the reviews and respond to those reviews, because there was a lot of things that we had to, not a lot of things, actually, we had only a handful of things, but we had to be very clear when we published what was what and, you know, what technique we used for what thing and that kind of thing.
Yeah, what's real versus what's stretching me.
Yeah,
to say the truth. But I think everybody knows what I'm getting at.
There's a few places where we actually, if you look in that paper, we say something a second time because the reviewer missed it the first time and they wanted to clarify it again. So we actually say it a second time.
Right.
All right. That's been a lot from Brian and I. Do you guys have questions for us?
Hey, this is Deb. I'm going to start with one of the first questions. One of the things I hear about often is that people are looking to be either first or last author. How does that work in this kind of relationship where there's so many authors and so much vying for that first and last spot?
Sure. So with children's, we've been, what will typically happen is that there will be a co-first author and a co-last author. And where we typically have gone when we collaborate with academia, we also have a relationship with VCU, right down there in Richmond, is we typically give the academic lab the first author, and we take second co-first author. and then we'll give the academic lab the last author often and take second to last author. At the end of the day, publishing for HemoShare is about getting the data out there to support our programs. Who the authors are and the order of the authors is not going to impact the success of what we do. But that is very important for Kim. I wouldn't say it's important for Marshall anymore. Marshall's very generous in terms of authorship, but for someone like Kim who's at a certain stage of her career, to be the first author or the last author, we know that's important. We have individuals at HemoShare who have been in academia and been there before, so we're pretty open about that, and we just want it to be the best for our academic collaborator.
And we have a tendency to have a conversation about that as we go along. So, you know, we just took 4 posters to SIMD.
Yep.
So 4 posters together to SIMD, which is the Society for Inherited Metabolic Disease, which was last week. And we have a conversation about who's authors on those posters and where we're placed. And then so that authorship may get moved around for publications a little bit. But, you know, it's things like that I looked at their posters, they got to see my poster, it's just kind of this back and forth.
And I just was texted. Can you tell us anything about any patents or how you have a conversation about patents? I'm not sure if you guys are there yet, but maybe you can comment from other relationships you might have.
Sure. So we are there at HemoShare in children's, and it's very clearly laid out in the original collaboration agreement that inventors, if they come from children's, are authors on the patent. So it's pretty clear. At the end of the day, just to talk a little bit about that, what you don't want to do, whether you're patenting something out of academia or whether you're patenting something out of industry or whether you're patenting something together, is you don't want to end up in a situation where you've omitted an author who could then invalidate the patent in litigation, meaning they should have been on there. You also don't want to have authors that shouldn't be on patents, but the real risk is when you overtly omit someone that should have been on there and then they take you to court on that. So, but it's something that, like, if you do it right from the get-go in your collaboration with industry, your tech transfer office will begin to lay all those rules out. And it's just also important to, you know, have an open discussions as new ideas arise in conversations. And, you know, if you're truly with a good collaborator, they're going to want you to be on those patents. It just strengthens the patents to have more of those experts on it that really contributed to it.
And as, you know, the academia person, it's bring it up early and have that conversation. At a certain point, I think Brian would agree, it's sort of out of our hands. It goes to the lawyers. And it's also talk in academia, talk to your tech transfer people as well, and whoever does your patents, so whether that's legal or otherwise, because that also, them being looped in earlier as opposed to later, will save you some headaches.
Yeah, it's also important that you know that so you may be on an original patent around some concept, but as a pharmaceutical company advances that work, it's likely that you won't be on those patents. For example, as it gets deep into chemistry and now there's modifications and all these chemical moieties that are being added to the baseline structure, the people that are on those patents are the chemists that do that. So, like, I wouldn't even find myself on a patent like that. And the reason, again, for that is you can completely invalidate a patent if you have someone on there who's claiming to have contributed to inventorship but didn't contribute to inventorship. So it's very different than when you publish a paper. I'm sure we've all been in situations where we're like a middle author because we contributed something to it. You know, we may have not written any of the paper, but we were involved in some of the conversations. So, yeah, you know, it's a very academic thing. We put you on as a middle author, and people know that, you know, when you're a middle author, you've done some things, but you clearly weren't the first or the last author, or the second to last author. Very different for a patent, though. Like, you have to really scrutinize every author that goes on there so that you don't invalidate that intellectual property, because it's that intellectual property that's necessary to ultimately, for example, if it's around a chemistry, without that intellectual property, you don't have the freedom to operate or the protection to take it into the clinic. Therefore, there's no market. Therefore, you're not going to have a product. So those are two very different worlds in terms of why authors are or are not on patents.
And it can take a while. Patents can take a while.
That was really helpful.
And authors can be added to patents afterwards. Often we will file patents here at HemoShear, and we'll just take the major inventor and maybe the second person involved, and we just get it filed because we know we need to get that provisional application out there. And then as time goes on, we'll do an assessment with our attorneys. Okay, so who else should be on there? And it's a very careful process.
That's great. That was really helpful. I have an interesting question here. This person's going to remain anonymous. When patients' data or samples are contributed, do patients share in any financial benefits, or how are they otherwise compensated or share in benefits of the resulting treatments?
Depends on the consent. So, so if you are consented to donate your sample, it depends entirely on the wording of the consent. So if the consent says you are not going to receive any or you are not going to receive any financial benefit from your donations or future discoveries based on this sample that you're providing, then you wouldn't participate in that. If your consent is different, so it has a lot to do with what you have as the original consent for the sample and the language that's in those consents. And I know everybody's scrambling to look at their IRB consents right now. So if you're writing the IRB consent, it depends what you label it as and where that sample comes from. So if you're the one in charge of it, it has to do with that part of your consent. Did that answer your question?
It does. Do you find...
Actually, sorry. Hi, this is Peter. I asked the question. I didn't need to be anonymous. But what I was thinking, so yes, I understand, the legal technicality. I'm thinking more in the sort of moral sense of, we want to, the patients, that we're doing this for we want them to benefit from whatever treatments or things ultimately come out of this. How, and we wouldn't be able to do this research without them. So, how do you take that into account when you're finally do develop a treatment and make it, and how can you make it available, as much as possible?
So there's no reason with the therapies that we designed that anyone that was in the cohort whose cells were provided would not be part of that. They may not be the first level of, so they, there's stages of clinical trials. There's 1, 2, 3. They wouldn't be in the first group. They may be later in the second group, so stage 2 trials, in part because they had a liver transplant, so they're going to be a little bit milder. Most of our patients get thanked. They get, they get acknowledged in all of our papers. They get acknowledged in all of our talks. A few of them actually know that their livers went here, and so we thank them pretty regularly in person. Brian, do you want to talk about that at all? Most of them are anonymous, so Brian doesn't know who they are. I know who some of them are.
Yes, I mean, we're very blessed that we have patients and families that after transplant are willing to essentially donate their liver to our research, whether it be research to better understand the disease research to develop a therapy that would ultimately be for them. You know, it's always our fullest intent when we're going into a disease area to advance a therapy with those patients in mind. So the ultimate reward for them and other patients like them is that we're successful and have a therapy that makes it through the rigors of clinical trial development. So I'm not sure if that answers your question. There are some instances, like for example, we've named rooms at HemoShare after our patients with the family's consent to do so. We even take it to the point that every patient's liver we receive, even though we don't know the patient's name, we give that liver a name, and then we treat every experiment with that patient's name. So they're all made up names, like Stacy, our very first patient, TJ, our second patient, and the list goes on and on. We've now done this. We've got probably close to 30 livers now in different inborn areas of metabolism. So, you know, we take it very seriously because it's an opportunity that you get, but there's also a lot that goes into us receiving a liver from them, both from an economic standpoint as well as just a human capital bandwidth standpoint. We've had to establish with Kim and Marshall, working very closely with us, a liver repository or a biorepository that now spans the entire country. It's not just Georgetown. There's Columbia, Mount Sinai, Pitt, Baylor, Seattle, Stanford, Utah. We're bringing others on all the time, but to do so, you know, we've had to create a human infrastructure to make sure that when a liver is excised from a patient, you know, what happens, if you've ever seen this in the transplant, in the room at the time of that, is everyone that's scrubbed in for that transplant, they're not worried about the liver coming out of the patient. It literally goes into a bucket. So we can't tap into that resource to have one of the nurses or surgical techs take that liver and go prepare it again. We have to create another infrastructure where someone can then receive that liver, perfuse that liver as if it were going to be transplanted into another individual, even though it's not, such that it can be stabilized, so then the liver can either be transferred to HemoShare to do the cell isolation here, or if there's a facility close or even within that transplant center that can do the isolation, they will do the isolation. But coordinating this all is challenging because the team that's doing the transplant, what they're most concerned about and the thing they should be most concerned about is that patient on the table who's being transplanted, not their medical waste that's coming to us. So there's a lot that goes into this, and I think, you know, we've learned a lot over the past 2 years. Our first transplant was July 10th, 2015 at Georgetown. Like I said, we've done a significant number since then, and even considering expanding it into Spain, where there's a liver isolation facility there for Europe, the Middle East, into King Faisal Hospital, Saudi Arabia, and down to Garan, down in Buenos Aires in South America. The idea being that this precious tissue, which contains cells in a disease state, you know, often animal models don't mimic the human disease. It's a great resource, not just for HemoShare, but we share these cells with academic facilities across the country at no charge.
Hey, thank you so much. That's great. Thank you so much.
You're welcome.
And I'm just thinking of, you know, when I'm talking to a family or a patient, you know, and, you know, I'm sure they don't, I didn't even realize how much went into all of this, but, you know, they want to ultimately know, I think, that they're helping other patients, I think, and so that's helpful to know sort of how the whole process ultimately produces, hopefully, treatments.
That's great. No, thanks for asking.
This is Deb, and I'm just going to anonymously say that maybe I saw a Western blot at the meeting and I knew it was one of my patients. And maybe I took a picture of the one lane on the Western blot so I could email the family and say, I just wanted you to know that I know who this liver came from and it's the one I know you love, and thank you for supporting research. And it was just, you know, the one lane on the Western blot that I knew was her. And I got the sweetest email back saying, we're so happy she's helping other children. It's so important to us that you did that. Thank you for protecting her liver and helping other kids. So I think that this whole idea of can we somehow, at least in the same way that that you're doing, Dr. Wamoff, this idea of how do we kind of tell the patients this might not help your child, but it's helping all of society, and giving them that support back is really helpful too, even if it's not directly a medication for their child.
Wow, that's great. I'll share that story with our entire team. It's stuff like that kind of why everybody gets out. You know, if you were to ever visit HemoShare, and everyone on the phone is always welcome to come down. I'm not sure if you're all based at Children's or in the DC area, but when you walk down the hallways of HemoShare, we don't know the children's name, but there are pictures of Kim's patients and other collaborators' patients lacing the hallway. And we may or may not have received one of their livers. We'll never know, but these are kids who are really sick that highly motivate everyone around us. And those little stories about a Western blot, I mean, you know, when you're a scientist, those Western blots mean a lot to you. You know, people may joke about, oh, it's just another piece of data, but, you know what goes into generating that, and to see that connection go back to the family and now back to us on the phone, that's pretty cool. Thanks, Deb.
Yeah, I'm gonna go here briefly. It's not a biorepository talk, but Kim, do you just want to give us a minute or two on the usefulness of a biorepository or something like that in building these relationships, and is that needed for this type of relationship, or does it at least help this type of relationship? Hey, Kim, we can't hear you. Hold on. Can you guys hear Kim?
I cannot hear Kim.
I'm going to go pull her into my office. Hold on. Brian, you want to talk about that's the right question. I'll grab Kim.
I'm trying to, yeah, so the importance of a biorepository, was that the question?
Yeah, tell me if you think the biorepository is why this worked as well as it did, and is that really needed if you're going to do this type of relationship? And would that be a place to start, even if you're not sure where the research is going to go right now?
Well, yeah, so we didn't create a biorepository for the sake of creating a biorepository. We needed a biorepository for these drug discovery programs and other drug discovery programs to work. The challenge that we had as we got into inborn errors in metabolism is that often the mouse models are very limited. With PA, propionic acidemia, and methylmalonic acidemia, without some genetic tricks, most of those animal models are embryonic lethal or neonatal lethal. So that told us that, okay, and you're also dealing with mouse models, whereas, you know, in branched chain amino acid catabolism, very different in mice than in humans. I mean, there's enzymes that are constitutively active in mice in those pathways that are inactive in human normal branched chain amino acid catabolism. So then it came to, well, you know, this is an area of high unmet need with a lot of patient pain. How are we going to do this? And that's where Marshall, kind of the light bulb went off in his head. He said, well, wait a second. There's kids that are transplanted, and for all intents and purposes, their livers are actually healthy. It's just that they can't break down these essential amino acids, and that's what's developing toxins. So there wouldn't be any challenges in trying to work with these cells. It's not like they're sick. It was just we needed to get the cells. And that's how we ended up building the biorepository. Now, there's other diseases that are not in the liver that we're working on, diseases in the vasculature, where very challenging to get human arteries, particularly from children with these vascular diseases. So we're taking a very different approach there. We're collaborating with NIH, NHLBI at NIH Intramural, where they're generating iPSCs from these patients with these vascular diseases, and then they're differentiating them into endothelial cells and into smooth muscle cells. And then from there, we can use them. So a very different approach. So I think before you would ever go down this process, because there is a ton of paperwork and IRB work that needs to go into place, you have to ask yourself, what is the best model for you to move your program forward? And there's some diseases where there's pretty good mouse models out there, and you may not need to go through these heroic efforts like we have with these diseases.
But I think since we're a rare disease group, I think thinking about rare disease, your patient's samples, whether it's blood or explanted organs or autopsy specimens or skin, depending on what you do, those folks have that kind of sample in a lab. You just can't recapitulate. I mean, you can talk about using CRISPR and you can talk about using a lot of other things, but I think a biorepository for most diseases is really useful. Whether or not you're always going to need that for your industry partners is a different issue, but I think when you work in rare disease, samples from your patients, even if it's the leftover blood from their usual blood draws, could be beneficial to someone. So our biorepository isn't just organs, it's actually also extra blood, and it's DNA, and it's skin tissue, and it's urine. And so at some point, that plasma or those PBCs are going to be useful to you or to somebody following you. The biggest pain, as Brian probably alluded to, is an IRB, because you need an IRB that's broad enough for many people to do many different types of experiments, but doesn't have a consent form that is overwhelming. And one of the problems with the biorepository is you don't know exactly what all of the samples are going to be used for. The other thing that can be daunting is space. So where are you going to put the samples that you have? Now, there's a number of great national biorepositories out there, and depending on your disorder, there may be a national biorepository. But that's really handy when you have one.
So that leads me into my homework assignment.
Well, so that's a really good, just real quick. You brought up some really good points, because if the research you're doing in your lab is you ultimately want to get to a point where you've got a molecule or something that could be a therapeutic, or you want to collaborate with a biotechnology company to bring a therapeutic forward, having human tissue at some point during that program is going to be important, because you want to know in terms of target validation and then target engagement of whatever your molecular drug asset is. You ideally want to do that in human patient samples, which could be as simple as PBMCs. So, I'll just kind of step back. Depending on where your program's going and what your intents are, getting to human tissue will ultimately be important for a drug discovery program, without a doubt.
All right. Yeah, I want to make sure that everyone on this call knows that many of the RDCRNs have a biorepository. So if you don't know if your group has one, this would be something you should ask that mentor of yours. They might have a biorepository that either you could get samples into for your patients that might want to give back to science, or that you might have access to if you have an experiment that might fit the requirements of that biorepository.
And some of the big institutions actually have some biorepositories as well, which may also be a sample.
And there are infrequently families that say no to biorepositories, but more often it's a yes. And the infrequent times when it is a no, I think that families always feel a little bad saying no to us, and giving them the space to say no is very appropriate. And so I'll be honest, with children that are dying, since that's what I tend to work with, I've had families that have viewed it as a gift that their child gives, and other families that couldn't imagine me taking a piece of skin from their baby after they died. So having that respectful conversation is actually important, and having that conversation early helps, like before we're at imminent death. But something like a transplant, I think, is ideal, where it's tissue that you would be losing anyway. That's a much easier conversation. So think about those things.
And I think when you're talking about getting consent for these things, always leave them the opportunity to say no. So what often happens is I'll kind of introduce the biorepository, and then Deb will go in and actually do the consent with the family.
Or let them out of it.
Or let them out of it. Mostly because I also happen to be the PI on our biorepository. So sometimes people are afraid to tell me no, but aren't afraid to tell Deb no.
Yeah, I get told no all the time.
Yeah, I know.
Yeah.
Or also, sometimes the people that actually will donate to the biorepository are not the people that I was expecting to. That I was not expecting them to donate. It's families where you're like,
really?
Really?
Yes.
You say no to everything else. Okay. So that's the other thing to know.
Well, this has been a really great call. Thank you, Kim. Thank you, Brian Wamoff. I can't say again how grateful we are to have this conversation with the two of you and to hear your perspectives. As always, anyone on the clinical research group, if you have more questions, I'm happy to forward those on to either Dr. Wamoff or Dr. Chapman if there's something particular. Always feel free to text or call me if you have questions that come up. And thanks so much for joining us on this, at least here in DC, cold, rainy, and possibly snowy day. So thanks all and have a great week. Thank you. Bye.
Bye-bye.
Bye.
Bye.